Welcome back, everybody. We will now start our second session by welcoming Dr. Damla Beton, who will present her project named Stable Isotope Analysis as a Tool for Conservation Biology.
Hello, my name is Damla Beton, and I'm an independent researcher at SPOT. SPOT is a part of Marine Turtle Conservation Project, which is a collaborative approach between SPOT, the University of Exeter in UK, and TRNC Ministry of Tourism and Natural Resources. And today I'm gonna give you a talk about stable isotope analysis as a tool for nature conservation, giving an example through Marine Turtle Conservation Project. There are 8 species of sea turtles in the world. 2 are found nesting in Cyprus, the green turtle and the loggerhead turtle. Our project has been studying them for 30 years now. Let me give you some background about the life cycle that will be important to understand for my talk. Let's start at the bottom of the life cycle diagram on the right of my slide. A turtle is on the beach, let's say in Cyprus in June. She lays around 100 eggs and goes back to the water, but through that summer she stays near the beach and every 2 weeks comes back and lays another nest on the beach. At the end of the summer, after laying 3 to 5 nests, she leaves Cyprus and migrates to foraging grounds that may be thousands of kilometers away. Meanwhile, hatchlings from the nests enter the sea and they are carried by the currents, developing an open ocean for around 10 to 30 years. Not much is known about this period, and so they call it the lost years. But when they are mature, they join the rest of the adults in the coastal foraging grounds. Every 2 to 8 years, the adults migrate to Cyprus to breed and lay eggs. They do not lay eggs every year, so you can see that it is a complicated life cycle, and spending their lives in many different habitats all across Mediterranean makes them vulnerable to human threats at any of those stages. They use the same foraging sites and nesting sites year after year. We know this because we have been satellite tracking them for more than 20 years. In the picture, you can see a turtle that has a satellite transmitter attached to its shell. In fact, every turtle that has ever laid a nest at Alagadi since 1992 has been tagged with a flipper tag and a microchip. Some of them we know very well indeed. In 2007, we reported on 5 individuals that we managed to track on more than one occasion over many years. All of them showed high fidelity of nesting beaches, migration routes, and foraging sites. You can see the gray migration route from their first satellite tracking and the black line from when we tracked them again a few years later when they returned to Alagadi to breed. From nest counts and beach patterns, we know where the turtles are nesting, and the government has put the most important nesting beaches under protection with a network of special environmental protection areas. At all of these beaches, teams of volunteers check every day for turtle nests. When they find them, they protect them with a metal cage that allows hatchlings to escape but prevents dogs and foxes, which are introduced to Cyprus by humans, from digging into the nest to eat the eggs. Because of this action, thousands more hatchlings are raised successfully. And because of the nest protection, the population of turtles started to increase in around 2009. So almost 20 years after nest protection began, those saved hatchlings were observed returning to Alagadi, where the proportion of neophytes, which means the young turtles that are new to the population and without tags, from previous nesting years became dominant in the population. So this gave an indication that the population was responding well to our long-term nesting caging efforts. We did a good job at the nesting beaches, but they may still be a threat in foraging grounds. If they are all foraging in different countries, how do we decide which feeding site to protect? Between 1998 and 2011, we tracked 34 green turtles from 4 countries. 22 were from our project in TRNC, which led this work through Exeter PhD student Kimberly Stokes, who published a study in the journal Diversity and Distribution. 10 foraging sites were discovered. 50% of the tracked turtles used 2 sites in Libya, Bomba and Sirte. So was the case closed? No. We spent 14 years on that tracking. Many volunteers and staff worked on the beaches to attach the tags. Each satellite tag costs £2,000, and you pay an extra £1,000 per turtle for the satellite data. Just for the equipment and data, it costs over 1 million Turkish lira to fund that work. Yet the turtles tracked represent about 0.01% of the population. This might not be representative. Stable isotope ratios can be used in ecological studies to understand where animals are foraging through the concept that you are what you eat. Let's take carbon as an example. In the environment, it can be found in 2 forms, carbon-12 and carbon-13. Carbon-13 has an extra neutron in its atom, and it makes it more reactive and less stable. Depending on the environmental conditions, the reaction rates will vary, causing detectable differences in the ratio of carbon-13 to carbon-12 in different regions. So in 2015, we set out to understand with certainty where our green turtles were mostly foraging, using samples collected for a genetic study that we decided to apply to stable isotope analysis. This work was carried out by University of Exeter PhD student Phil Bradshaw and was published in the journal Marine Ecology Progress Series. Here you can see the nest count data for green turtles for northern Cyprus study beaches, with year on the x-axis. The red box shows the period during which the 2015 Kim Stokes tracking study was attaching satellite transmitters. The blue box shows the period during which we were taking samples from turtles that we later applied to stable isotope analysis. The blue arrow indicates the period from the initiation of the nesting protection to the start of the positive conservation response that was driven by the neophyte recruitment. And these slides show the situation on the beach with sampling and the kind of sample taken, a small piece of skin. To begin with, we looked at the carbon-13 and nitrogen-15 and compared stable isotope ratios of turtles that Kim Stokes had satellite tracked, represented by the dark field shapes, and for which we knew the foraging site, with samples from the rest of the population, represented by the open circles. There were clear clusters that we could use to assign likely foraging sites of the untracked individuals. However, one interesting cluster stood out, and we did not have any tracked turtles with which to reference to that cluster to a geographical location. So Phil Bradshaw developed a hit list of 48 turtles from the Alagadi database, whose M15 and C13 values were at the center of this cluster. The field team carried a list of the flipper tag numbers, and we chose 5 of the target turtles to track. I believe this is the first and perhaps the only example of an animal tracking study where such detailed analysis have been carried out to put the plan and track them. All 5 of the target turtles went to Bardavil Lake in Egypt. Our pre-2011 tracking studies missed the important side because back then it was less important before the population recovery. The numbers in the black circles show how many of the 27 tracked turtles were tracked to each site. But look at the pie chart and you see that Lake Bardavil in Egypt is by far the most important to the population. When the stable isotope analysis of 230 individuals is considered, as 40% of all the turtles are assigned to that location. So through stable isotope analysis, it emerged that the population recovery of the Mediterranean green turtle was linked to Lake Bardavil and may not be entirely due to nest protection. Lake Bardavil has undergone some changes in the last 30 years, with new entrances built which may have increased its suitability as a foraging site. To have so many turtles dependent on just one site makes the population extremely vulnerable to human changes such as fisheries pressure and climate change. In 2018 and 2019, we tracked 20 turtles from the Karpas Peninsula, another major nesting site. And 70% of the turtles used Bardavil. So as the population continues to increase, we see a greater proportion of turtles using the site. Stable isotope analysis allowed us to correct our satellite tracking data. On its own, satellite tracking is too expensive and doesn't provide certainty. Combining with SIA on rest of population, much more powerful. Although green turtles are recovering rapidly now, most of them are foraging in Bardavil Lake. Publication in scientific literature is key to ensuring impact in other countries and developing international collaboration needed to protect sea turtles. Thank you for listening. Please feel free to send us any questions you have to the email address mentioned here, info@cyprusturtles.org. And please follow our Facebook page called SPOT, Kuzey Kıbrıs Kaplumbağalar Koruma Cemiyeti. Soon we will be announcing a call for volunteers for the summer 2021. And please apply if you're interested. Thank you. Have a nice day.
Thank you, Dr. Beton, for this wonderful presentation. I would like to welcome Dr. Zeynep Koçer, who will now give her presentation on a very timely and significant topic, influenza viruses with pandemic potential. Welcome, Dr. Zeynep.
Thank you so much for the invite. Let me put my presentation up first. Can you see my screen? Hello?
Yes, Hocam, we can see it.
Yeah, you can see it now?
Yes.
Okay. Well, thank you for the kind invitation and also a kind introduction. As you said, my name is Zeynep Koçer. I am working as an assistant professor at 9 Eylül University, İzmir International Biomedicine and Genome Institute. Also, I hold the principal investigator position at İzmir Biomedicine and Genome Center. I work as a research group leader at Emerging Viral Diseases Laboratory. Besides these duties, I also hold the chair position for the Biosafety Committee at IBG. And also I am doing I'm the director of the ABSL3 platform. So today I'm going to talk about influenza viruses with pandemic potential. I think there's a delay in pressing my slides. Do you see any movement in my slides? Because I don't see anything.
No, hocam, we can't see any like movement. It's still the same.
Okay.
First one.
Yeah, it happened the other day too. So let's give it a second. No, okay. Now I think it's moving, right?
Yes, it's moving.
Okay, I think there's a delay. So before I start talking about influenza with pandemic potential, I would like to introduce you to the One World, One Health terminology. So this means that, we as human, we need to work with animal and plants and environment happily together to sustain a healthier place in this world. So when we look at the nature, we know that 60 to 75% of known and emerging infectious diseases in human arise from domestic or wild animals. Therefore, it's quite crucial to assess the health risks at the human, animal, and environment interface so that we can develop proper action plans for the veterinary and public health. When we look at the nature, we know that there are a lot of zoonotic pathogens, but I'm going to focus on the zoonotic viruses because virology is my field of work. As you know, Ebola, Marburg, Lassa, Hanta, Crimean Congo, which is quite common in Turkey, Nipah, Hendra, rabies, West Nile virus, influenza, and now coronavirus, and actually multiple types of coronaviruses. And there are so many other zoonotic viruses are available in nature. So these viruses are generally live in the bodies of different animals, but with one little mutation, they can transmit to mammals, including humans. Therefore, we always have this pandemic risk. And actually, in May 2017, the Time magazine made its cover just like this, warning, we are not ready for the next pandemic. Yet, in 2020, March 11th, WHO announced a new pandemic from coronavirus, which was a novel strain of coronavirus. And eventually it was named as SARS-CoV-2 to differentiate it from the previous SARS-CoV outbreak. So since then, actually, when we look at the numbers, you see here, let me put my pointer too. Okay, you see the global deaths here. It's almost 3 million people lost their lives in a little over 1 year. And here you see the number of cases around the world. And Turkey is quite up on the list in the world right now. So these numbers are changing every day. And this is the numbers just I took this from the site a little before the presentation, actually. So, well, I know we are all bored of SARS-CoV-2 pandemic at the moment, so I'm going to switch the gear and I'm going to talk about flu pandemics instead. So, in the recorded human history, we have 4 known flu pandemics caused by different subtypes and different strains of influenza. The first one appeared in 1918 from H1N1 virus. And then in 1957, we had H2N2 pandemic. And in 1968, we had H3N2 pandemic. And then finally, in 2009, we had another pandemic caused by a novel strain of H1N1. First of all, let me introduce you to influenza viruses. They belong to Orthomyxoviridae family. And there are 4 types of influenza viruses. Influenza type A, B, C, and D. The pandemics are mostly caused by influenza A viruses because influenza A viruses can infect a variety of host organisms. They are zoonotic, which means they can transmit from animals to human, and they may cause pandemics. However, when you look at the nature, the influenza viruses are carried by these cute little birds, the charaodiforms and unciforms. Charaodiforms are generally shorebirds and gulls, and unciforms are ducks and geese. Like when there's always pandemic, there's a speculation that this virus is a lab made. However, if you ever watched the movie Contagion, which appeared on the movies, at the movies in 2011, there was a scene in that movie, and they were discussing if this is a terrorist attack or whatever, and if it was human-made virus, because the movie was about the pandemic as well. One character in that movie was saying, like, the birds are doing it for us. And actually, this is so true for influenza. The birds are doing it for us too. These viruses don't harm these birds generally. However, with little mutations, they can transmit to mammals, and also they can transmit to poultry, which becomes a serious problem. Influenza A subtypes are divided in different subtypes based on their hemagglutinin and neuraminidase protein. Those are two surface glycoproteins. Based on hemagglutinin protein, there are 16 types of influenza, and based on neuraminidase protein, there are 9 subtypes of influenza in birds, which means there are possibly 144 potential subtypes of influenza in nature in birds. On top of that, in 2012, CDC researchers identified 2 new subtypes of influenza in bats. They were quite different than the avian influenza. They identified these viruses based on molecular analysis. However, the virus isolation was not successful. As I mentioned, influenza viruses can infect a variety of host species. They are carried in shorebirds and waterfowl. However, from there, they can transmit to swine back and forth, and they can transmit to poultry back and forth. Some strains can transmit to horse, from horse to dog, and some subtypes can transmit to sea mammals. And from swine also, the virus can jump to humans. Sometimes from avian species, it can jump to human. And from human, we can also infect our domestic cats and dogs. And also the virus can be deposited in abiotic sources. So when you look at it, the danger is everywhere. The virus causes asymptomatic gastroenteric disease in avian species, therefore it doesn't harm these birds. Maybe they have a little diarrhea, but that's all. However, the virus causes respiratory tract infection in mammals. And this is all because of binding a different type of sialic acid receptors found in different organs. For an avian influenza virus to infect a mammalian species, it needs to have a unique gene constellation so that it can facilitate successful zoonotic transmission. Therefore, the environmental stability of the virus is quite important. The adaptation success rate is quite important. After passing into the new host species, the replication ability and replication efficiency of the virus is quite important. Once it can replicate in the new host, the transmission efficiency between that new host members are also quite important. And this is entirely called host switch. The genome of the virus is a negative sense single-stranded RNA genome. It's quite similar to coronavirus, but also different. Coronaviruses are positive sense single-stranded RNA genome. Also, the difference of influenza from corona is influenza has a segmented genome, which means it has 8 gene segments, it synthesizes them separately, and then it packages them all together. But these 8 gene segments also codes up to 10 to 12 proteins with some accessory proteins, as you see here. Because of all these fancy genome structure, the virus can go through rapid genetic changes called genetic drift and genetic shift that I'm going to explain in a little bit. One important genetic change is point mutation. Because the virus has an RNA genome, the genome is synthesized by RNA-dependent RNA polymerase, which lacks proofreading ability. So once the genome is synthesized, the enzyme doesn't go back and looks at the error rate, and it doesn't try to fix those errors, and the mutation rate is higher in RNA viruses. And these mutations can occur in any gene at any site. For example, like when we talk about low pathogenic avian influenza viruses, the hemagglutinin protein needs to be cleaved, just like SARS-CoV-2 virus's spike protein. So where the protein cleaves, there is this motif of GLF, and right before the cleavage site, low pathogenic avian influenza viruses contain an arginine, only one basic amino acid. However, once this low pathogenic avian influenza virus adapts to poultry species, then the virus acquires multiple basic amino acids at the cleavage site, which makes them highly pathogenic. Because then the hemagglutinin protein can be cleaved in multiple organs, and the infection becomes systemic, and generally the infection kills the bird in 24 hours or sometimes less than 24 hours. Therefore, when they detect high pathogenic avian influenza viruses in poultry, the only way, unfortunately, is cooling the animal at the entire farm, because you cannot identify the disease in individuals in that limited time. Luckily, we don't have any human-to-human transmission so far from these high-path viruses. That kind of is a relief for us. It doesn't cause a pandemic. But another important genetic change in the genome is the reassortment, or genetic shift that we call. This happens when a host cell is infected with multiple strains of influenza at the same time. Then when these gene segments are synthesized in the host cell, they can package accidentally together, and this accidental packaging causes a novel virus. We call them reassortant virus. And most of the time, actually, the reassortant virus is the one causing the pandemic. For example, in 2013, one novel H7N9 virus appeared, and this novel H7N9 virus acquired its gene segments from domestic ducks, from the viruses circulating in domestic ducks, from the viruses circulating in wild birds, and also H9N2 viruses circulating in domestic poultry. Actually, these H9N2 viruses circulate in poultry most of the time in many countries, but they're low pathogenic and it doesn't hurt anything, except maybe the egg fertilization rate or whatsoever. However, when they donate their internal gene to other viruses, this brings the danger together. So in 2013, we had this H7N9 virus due to multiple reassortment events. And then, unfortunately, this virus was able to transmit to human and caused fatalities in almost 30% of the patients that were infected with this virus. And sometimes we see other outbreaks happening in different parts of the world. One virus was H10N8. Sometimes we see this randomly and occasionally happening. Again, in 2013, one lady who was 73-year-old was infected with H10N8 virus, probably coming from poultry, because she had the contact with live poultry market. This was quite interesting story from, again, like, avian influenza viruses. In 2016, one cat in New York shelter was infected with influenza, and then the virus was identified as H7N2. However, this flu strain was also able to transmit among cats, and it infected 45 cats in one shelter. And more importantly, one veterinarian who take care of sick cats also got infected with this virus. So the story here, probably one cat ate a bird who carried this avian influenza virus, and the virus was able to infect the mammalian cat, and then also it was able to transmit between different cats, and on top of that, it was able to jump to human. So, but luckily, this was a local outbreak, and they immediately stopped it at its place, and we didn't have any further problem with that. So let me talk about the pandemics a little bit in detail. In 1918, as I said, we had a pandemic caused by H1N1. This virus was fully avian origin based on some scientists, or some other scientists thought that the virus was a reassortment of mammalian viruses and previously circulating human strain. Because there is no solid decision on this, the virus was isolated back in time, from a person who got infected with this virus and then died in 1918, was buried under a glacier, and they found this body later on, and then they isolated the virus from that body that was frozen years ago. That's why the origin of the virus is still not known. However, what we know is the virus was emerged in a military camp in Kansas, USA, although it's called Spanish flu. And then it was spread to other countries by the deployment of soldiers during World War I. However, if you think about the human population in the world in 1918, this virus caused more than 50 million fatalities worldwide. This is a huge number. Actually, it's kind of like cleaning the world population, basically. Then in 1957, we had Asian flu, H2N2. This virus emerged in Singapore, spread to Hong Kong and USA. It acquired its 3 genes from avian influenza A viruses. Also, this virus caused 1.1 million fatalities worldwide. And this is also an interesting case. This H2N2 virus appeared in 1957 until the next pandemic, 1968, with H3N2. This H2N2 virus donated 6 genes to this new pandemic strain, and the other 2 genes came from an avian influenza A viruses. And then this H2N2 virus completely disappeared from the population, although rarely it's seen in birds still. The virus caused 1 million fatalities worldwide after it emerged in South Asia through Vietnam War veterans, again, and it was spread to multiple countries. So therefore, because none of us have immunity to this virus anymore, this is also quite a good danger waiting for us along the way. Hopefully that it's not going to pop up again in the future. Then in 2009, we had swine flu caused by a novel strain of H1N1. The virus was emerged in Mexico and United States and spread to multiple countries across the world. And it acquired its genes from 2 main sources. One of them was triple reassortant H3N2, and another source was Eurasian avian-like swine H1N1. However, as you see from the name, the one source was also a triple reassortant from avian H1N1, seasonal human H3N2, and classical swine H1N1. So basically what happened here, this virus went through multiple reassortment events over 20 years, and then it emerged in swine population and transmit to human population, causing more than almost 300,000 fatalities worldwide. So what do we do in our team regarding the pandemics? Our main research team is the emerging zoonotic viral diseases that can transmit from animals to humans. We look at the influenza viruses of avian origin, and also we have some projects studying the bat origin zoonotic viruses. Our approach is, first of all, surveillance, which means that sampling from environmental sources. And then we do the genomic and phylogenetic analysis to detect the critical mutations and also to determine the evolutionary processes in the formation of these novel strains. And then we do the virus isolation and characterization when possible. We cannot do the virus isolation for everything, but based on some legal permits or whatsoever. However, when we can, we do the isolation. Then our aim is to do the risk assessment by in vitro and in vivo assays. And our ultimate goal here is to monitor the emergence of novel zoonotic viruses and strains to perform risk assessment for public and veterinary health. So let me explain why such studies are important in Turkey, especially in terms of avian influenza A viruses, which I mean avian influenza A viruses, as I said, I mean 144 potential subtypes of avian influenza A viruses, not only the high pathogenic viruses. As I said, low pathogenic viruses are actually more important in the formation of pandemics. So when we look at the bird migratory routes, you see there are 9 major migratory routes for aquatic birds. And Turkey is located in the middle of Black Sea Mediterranean flyway. And also some branches of West Asian, East African flyway is passing here. And all these flyways are kind of overlapping at different locations of the world, giving the birds chance to mingle and also to exchange their viruses in the same environment. Here, because we are located in İzmir, maybe you already know that we have a bird paradise in Gediz Delta, which hosts thousands of migratory birds every year. But besides these birds, in the same habitat, we have wild boars, golden jackal, red fox, wild cat, jungle cat, weasels, and wild horses. And also we have a lot of people, human visitors in the area for some sports or leisure activities. So if you recall, this is quite similar to the host range of influenza that we can find almost everything influenza can infect in that environment. So in my lab, 2 of my students, Öykü and Emre, they are doing the study to identify the influenza viruses from shorebirds and waterfowls. And then my other student, Ekin, did his master's study to identify the influenza A virus persistence in abiotic sources, especially in sediment samples. But now he completed his thesis study and he started his PhD. So his PhD thesis is going to be in a different topic. And Öykü is also working on a separate project looking at the zoonotic viruses in bats. And my other student, Mert, is looking at the influenza A viruses and the genetic changes that we observe in humans during seasonal influenza infections. But today we are talking about influenza A viruses of avian origin. I'm going to show you a couple of slides regarding that work. First of all, we go out in the field and we collect our environmental sampling, as I said. This is part of the surveillance study. Once we collect our samples, we store them at cold temperatures until we get back to the laboratory. And then we do the molecular analysis, first of all, to identify if the samples are virus positive or not. Then we do the further analysis from there. Here, this is Yavuz, my former master's student. In his thesis, we collected 500 fecal samples from wild aquatic birds, and we found 156 influenza A positive samples by PCR. Then we did the virus isolation. And here you see hemagglutination assay, which means that after we inoculate the samples in embryonated chicken eggs, we collect the allantoic fluids of those chickens. chicken eggs, embryonated chicken eggs. Then we add chicken red blood cells on top of the sample. If there's any virus in the sample, then the virus is attached to red blood cells and avoid them to precipitate. However, if there is no virus in the sample, you see this, the red blood cells can precipitate to the bottom of the plate. So here you see the positive sample. And now once we look at this positive sample, we identify the subtype as H6N2. And actually, our paper just came out this Friday. It's online now. You can go ahead and read it if you like these type of studies. Also, in that study, we identified almost 30 mutations in the genome of the H6N2 viruses that were previously identified in the literature related to increased polymerase activity or, increased transmission efficiency or replication efficiency in mammals. So that's why those mutations are quite important. But here, for example, I'm just going to talk about one single gene. We observed mutations in every gene. In PB1, polymerase basic 1 protein, it's part of the polymerase complex of the virus. We identified some known mutations such as N375, P13. These are quite important mutations in influenza world in terms of host adaptation and host jump. Also in PB1F2 accessory proteins, we observed this S66 mutation in the genome of H6N2 virus. This mutation was associated with the increased virulence in mammals. This mutation is a very, very well-known marker, which was also observed in all pandemic viruses so far. So when he looked at the origin of the gene segments of the isolates, H6N2 isolate from Turkey, we saw that the PB2 gene segment was originated from viruses circulating in Ukraine. PB1 and PA were coming from Mongolia. And HA and NS gene segments were coming from Georgia. NP and NA gene segments were similar to the viruses circulating in Netherlands, and matrix gene segment was more similar to the viruses circulating in Egypt. So if you see, like this virus was basically the reassortment of multiple viruses circulating in the birds over this area in Asia, Europe, and Turkey. Then we took our virus genome and we did a study with, in a collaboration with our Ugandan and Egyptian partners because they isolated H6N2 virus around the same time in the same year as well. Then when we look at the genome of these viruses, here you see GD18 is our Turkish isolate. The viruses with EG, two of them, they're from Egypt, and UG731 is from Uganda. We identified mutations, common mutations in all these 4 viruses. They are shown in red. Also, we identified some mutations. They were seen in several of these viruses, or only in one of them. They were identified in different colors. So the number of these mutations are quite high, actually, which puts this virus as a kind of like a further study focus, because we need to look at the in vitro and in vivo evaluation of the risk assessment of these viruses circulating in nature in terms of their pandemic potential. So, and then we did the phylogenetic analysis to look at the relation of these gene segments with each other between 4 viruses isolated in Uganda, Egypt, and Turkey. Some of them, for Egyptian and Turkey viruses, actually, these gene segments were quite similar to each other, except NP protein, nucleoprotein protein. However, their HA was quite similar for all 4 viruses. So this gives us a kind of reassortment. Instead of showing you the phylogenetic tree, I prefer showing you this reassortment table. So you see the similarities and differences. This is how we identify the genetic constellation of the viruses. So therefore, when we look at the things in terms of H6 viruses, sometimes we see in either in the news or in the literature that, you know, this new strain of bird flu, it comes from, you know, the birds and infects humans. Also, there were some studies showing human infection with avian influenza H6N1, showing the serological appearance of the virus in human. And then also there's another study showing seropositivity for avian influenza H6 viruses among humans in China. So all these things bring if this virus has a pandemic potential or not. So nowadays our TÜBİTAK project was just approved and it just started actually. Now we are going to start our field work at the end of this month. So in this project, I mean, so far we did our study in Gediz Delta area here near İzmir. But in this TÜBİTAK project, we are expanding our research to the entire Turkey. So we'll sample 8 places in Aegean side coast of Turkey. And then also we'll collect samples from Salt Lake, and we'll collect some samples from Mediterranean coast, Black Sea coast, and also the eastern part of Turkey to sample almost all migratory routes passing through Turkey. And we will do this sampling for 2 years back to back, and hoping to identify more viruses so that we know if there's anything dangerous coming in our country or not, and given a timely response to that. In our another study, we do the zoonotic, we look at the zoonotic viruses in bats, and here I'm going to show you a couple pictures from the field work of it. We haven't started the laboratory work for that project. However, we did the field studies. It's quite a teamwork, and because you work with bats, you need to do it at night when they start waking up and getting out of the cave. So basically, this project is in collaboration with EcoHealth Alliance, which is a nonprofit organization. They support our field studies under Western Asia Bat Research Network. Also, there's Rashid Bilgin from Boğaziçi University, and also we join these field works. What happens is you put the nest at the entrance of the cave, and when they wake up, as I said, they try to get out, and then you can collect them from this net here. They don't get any harm because also they're conserved species anyway. You need to be working with them very carefully. So then we collect them one by one and put them in individual bags. They can also get air through these bags. They don't suffocate in it. And then we start collecting. This is our little lab area out in the field. Basically, we collect samples from birds, such as oral swabs like this. And after we collect all the samples from different bat species, we leave them out in the world. They fly out and get their food and then go back to their cave. So basically, to finish my talk, if you look at the Spanish flu, like, you know, we had more than 50 million fatalities. And with Asian flu, we had almost 1 to 4 million fatalities based on WHO's estimates. And then for Hong Kong flu, again, like we lost more than 1 million people, could be up to 4 million according to WHO again. And with swine flu, we lost almost 300,000 people's life. H5N1 outbreaks only, that high pathogenic H1N1 outbreaks caused more than 50% fatalities in the infected people. H7N9 avian influenza virus caused 30% mortality in the infected people. You know, COVID-19 is more than 2.5 million in 1 year. So on top of human life, all these, you know, outbreaks and pandemics causes millions of birds to be killed at the farms. And recently, due to COVID-19 infection in a mink farm in Netherlands, Denmark, there were millions of mink killed at the same time too. So when we think about all these mortalities of human and animal population, surveillance and risk assessment studies for zoonotic viruses are quite crucial, actually. So with that, I'd like to thank the Ministry of Agriculture and Forestry, different departments of it, for giving us legal permits for the field works and also for virus isolation and to work with these samples. I would also like to give special thanks to Emre Karayel and Özcan Alptekin, our drivers, to take us to fieldworks. Also, the National Park staff at İzmir Bird Paradise, they're quite helpful and they're always next to us when we go out in the field to protect us from any danger happening. I would like to thank the National Reference Laboratory at İzmir Borno Veterinary Control Institute for avian influenza. And then also I would like to thank our collaborators from different universities and different departments and from even different countries. So, but of course, my biggest thank goes to our funding agencies. The initial studies that I just showed you are in our paper published, was done by IBG Startup Fund when I started here almost 2 years ago. And also St. Jude Children's Research Hospital, which was founded by NIH, NIID Centers of Excellence for Influenza Research and Surveillance. This is the place where I did my postdoc also. I would also like to thank 9 Eylül scientific research programs for supporting us with a project. Also, I would like to thank TÜBİTAK for supporting our projects to do this type of work. But yeah, the biggest thank goes to my students because they do all this work in the lab. So I have a quite bright young people. I mean, this team of people are doing all the work that I presented here. So Ekin, Emre, Mert, and Öykü are my active students, and these girls were interns in my lab before. So with that, I would like to get your questions. Can I go back to stream page so that I can see?
Thank you, Dr. Koçer, for this very rich presentation.
I hope I was in a time limit.
Yeah, you were on the time limit, and we don't have any questions. Thank you for coming today.
Thank you.
Okay, now we're gonna go to have another presentations made by our graduate students. But before we continue, I would like to tell you guys, like, don't forget, please, to join Yakindor official channel, Yakindor University official channel on YouTube. Don't forget to subscribe there, and don't forget to follow ANU Genetic Student Association for any following events in the future. Now we're gonna start with a few presentations from the graduate students in Nearest University. So I would like to welcome Cansu Aydın. Hello, Cansu.
Hi, nice to see you.
Nice to see you too. Welcome.
Thank you. Can I start or? Okay, I can share my screen. Just give me a minute. Can you see my screen right now, or?
We can't see it yet.
Okay, wait. Okay. And now?
Yes, we can see it.
Okay. So, good afternoon, everyone. I hope everyone is fine. And first of all, I would like to thank the student community and our precious teachers for this organizing the amazing symposium. This is a very precious moment for me. So today I want to briefly tell you about what happened in my education life up to this time. So first of all, let me introduce myself. I am Can Saydın. I graduated in 2019. So that means I am the second graduate of this department. And now I'm doing my master's in medical biology at Trakya University. As of this semester, I am the thesis periods. Somehow I lived undergraduate life fully, even though we couldn't fit many activities events like that in these 4 years. There were times when I said that we did something when I looked back, and also it's amazing for me, these 4 years. And also I did my internship in Cyprus. I did it in Dr. Burhan Nalbantoğlu State Hospital in DNA and thalassemia laboratory. These places are special for me because these places became a turning point for me, because I was someone who was thinking of changing department until the 3rd year. I think I didn't understand what we were doing until then. Okay, I'm going to classes, I'm interested in the lessons, everything is fine, but something was missing. But when I smelled the lab environment for a long time, I said it to myself, yes, I should be here. So these 4 years have flown like water. I didn't know when we come to an end. Yes, it's a graduation day, but we were not lucky because there were some technical problems in the graduation hall. I guess it couldn't have been a worse graduation, but I think our closing, all kinds of negativity between us, as you can see all the photos, even though, and also this is our last meeting. Yes, there were some missing persons, but it was still a very nice night for us, especially for me. So yes, we saw so many lessons. We took many exams. We got sometimes angry, we laughed, and etc. But what about the next? What about our next step? My next step was clear because I had decided to what I want to do, what I want to be, what I wanted to work. If it was a little late, but it's okay. But some situations don't go the way you want. After the graduation, when I returned to Cyprus to close my house, we had a few meetings with Resim Hoca to see what we can do about my situation. She also offered me a few options for the next move, for my next move. I thought about these options and made a decision, but I had a little problem here. But that didn't daunt me. We evaluated this problem with another option, and I always wanted to be open to all possibilities and wanted to realize another option. So what was this another option for me, for my master or PhD level? My another option is Edirne. I found myself here in 2 days to be one step closer to my goals. I started my master degree here with a special student status. By the end of the first semester, I gained the right to be a student, and graduate students have the opportunity to work in the laboratory where their advisor is located. My first advisor was on working in the medical biology department. So I spent the first 5 months of my master's in the medical biology department. Projects were usually carried out in this department, like to-be-talk projects and etc. I learned so many different techniques here, like cell counting, passaging, phrasing, MTT assays, transval assay, wound healing, and etc. It goes like that. But my first advisor, we have some issue about my situation. So meanwhile, I had the chance to publish my thesis that I wrote during my undergraduate education. And again, I would like to thank Resmi Hoca once again for giving me this chance. If you are interested in epigenetic treatments for cancer, you can take a look at this article. With the end of the first semester, I decided to change my advisor because of some problems. I asked this to my old advisor. He accepted what I wanted, but it was not easy to change my advisor. It took me about 6 months. It means 1 semester. But at the end of these 6 months, I was able to change my advisor. At this point, I want to give you some advice. Please research your advisor at the beginning of the PhD or master level and choose an advisor who will be the most effective for you. As of August 2020, I found myself in the medical genetic departments. I'm working with my current advisor, which is Assistant Professor Dr. Emine Iqbal Atlı. She is the cytogenetics laboratory supervisor. There are genetic diseases diagnosis center and tissue typing laboratory in the Department of Medical Genetics, where I'm currently working. The activities of the genetic diseases diagnosis center consist of medical genetics polyclinic and the genetic counseling polyclinic, the molecular genetic laboratory, and the cytogenetic laboratory. So what are we doing here? We carry out many genetic diagnosis tests you learn during your undergraduate periods, mutation analysis, chromosome analysis, prenatal diagnosis, and many more. And, we learned some techniques for technical parts like FISH, NGS, or next generation sequencing, Western blotting, MLPA, RAC, GH. This is all about it. I have been working in the cytogenetics laboratory for 6 to 7 months. And about 2 months of this period, I have been working in the prenatal diagnosis laboratory. Here I am doing genetic analysis of patients by culturing amnions, CVS, and abortion materials that come to our department. These analyses require a bit of attention because you cannot ask the patients to bring back amnion and abort material. And my next step is the molecular genetics lab, where I can study my thesis. In my thesis, we plan to conduct genetic etiology study using NGS panels in non-obstructive azoospermia patients. So let's see what awaits me during the thesis periods. I have taken during my undergrad life, have provided my postgraduate education life. We have learned very valuable lessons such as cancer genetics, epigenetics, embryology, teratology, bioinformatics, ecology, nanotechnology, bioaesthetics, and etc. from really valuable teachers. That's why I want you to thank them in your presence. Yes, there were times when I rebelled, but looking back, it is good that I studied at this university, and I'm fortunate that I received training from such valuable professors. I hope you know and appreciate the value of your undergraduate life, because I can assure you that after my undergraduate education, I clearly noticed that the difference between me and my friends who study at different university. Yes, I know I am at the very beginning of the road. I know that, but also know that what I want. Of course, it took time for me to be so sure of myself. After being sure, I determined a road for myself. So the first step of this road was to be sure of my goals. The second step is to plan my goals step by step. But I did not constrain these plans to a certain period of time. Then I start working harder for my goals, and I stick to both my work routine and my goals. As I said, I am at the very beginning of the road, but you know, I believe that I will reach my goals at the end of this long and harsh road. There is a saying I love very much: if you woke up without a goal, go back to sleep. That's true. That's really, really true. There is no such thing as impossible in my literature, because everything is possible for me. And I hope you also know what you want from life and can do whatever is necessary for it, because everyone is the creator of their own life. So what can I say? I can say that for you guys, then stick to your plan, your goals, or whatever. So then come easy for you on this very long road. So thank you for your precious time, and thank you for your attention.
Thank you, Cansu, for sharing your experience, your wonderful experience with us.
Thank you.
I really hope you get what you want in the future.
I hope.
And get into the molecular genetics lab. Yeah. Thank you so much. No problem. Now we're gonna be with our second presenter, our second graduate student, Furkan Ayberk Binbay.
Hello, can you hear me?
Welcome, Furkan.
Hello. So, hello everyone. First of all, I hope everybody is well, strong and healthy. I do not have a presentation, but I have lots of stories to tell you. So, if you want, I can start. So guys, I'm Furkan Ayberk Binbay. As you know, I was graduate from the nearest university's Molecular Biology and Genetics Department in 2019, like Cansu. And just like you, I also sat the chairs you are sitting on currently. So I'm currently taking the Life Science Informatics Master Program at the Bonn Aachen International Center for Information Technology, which has been established for major institutions, Fraunhofer, Universität Bonn, Universität Aachen, and the Life and Medical Sciences. So in this institution, actually, I and my friends, my classmates, are working with a very tough group of academicians, you know. However, this is not a bad thing, you know. On the contrary, it's quite beneficial for us, because they are training us directly on how a real data scientist can manipulate a data set and make sense of it by using machine learning or deep learning methods. So actually, as we as bioinformatics candidates, we are trained for what we call problem solving, to solve various issues that can be encountered during a project. So I'm currently in the fourth semester, which is the last semester. And under this program, they give us the opportunity to specialize in two important, life science informatics branches. And one of them is the bioinformatics, the another one is the chemoinformatics. So of course, I'm here to be a bioinformatician, but actually, recently, I'm preparing myself to upcoming master thesis. Well, I will start to do my master thesis in the coming weeks, and the subject of my thesis actually touches on the both informatics field. But I'm going to I will work mostly in the structural bioinformatics because I'm going to model an inhibitor that should specifically bind to a specific subunit of the one of the best known membrane receptors. So my current goal will be to advance myself in the field of structural bioinformatics. During this process, I'm in the thesis process, and continue a career in this field after graduation. So of course, I have some advices for you, especially if there are people who among you who want to make a career in this field. I'm in the field of bioinformatics, so I'm ready to tell you about my experiences with bioinformatics. However, of course, I will not have the opportunity to explain everything to you due to the time restriction. But I will try to answer any questions you may have. So actually, if it's not from here, you can also send me a private message or mail. You don't need to be hesitant about it. And please feel free to ask any questions, okay, guys? So, well, it might be too nice to start with a common question, actually, that people often ask me. Sometimes people, especially my friends, ask me how I got my interest in bioinformatics. So my interest in bioinformatics actually started in my internship, So after discovering the techniques, or let's say methods, in different laboratories, I found it more appropriate for myself to do analysis in analyzing of the analysis of the data in front of a computer instead of doing the same routine things every day. Please do not misunderstand me, because I really miss working in the laboratories nowadays. So please don't get me wrong. However, well, this is the truth for me. Anyway, so after a while, I talked to the person at the head of the place where I did my internship about this thing. Well, he knew about me. He knew how was I working. And I told him that I want to take part in the analysis. And he also helped me to do this. And then maybe I just spent more than 1 month by analyzing the results coming from Sanger sequencing and NGS in various databases like OMIM, ClinVar, or Assemble, etc., and in silico databases like Mutation Taster, or by using specific softwares. And actually, at that point, I realized that what I'm doing is what really satisfies me. I'm in the data analysis. And actually, that internship was a turning point for my future career. Actually, in addition to my internship, several courses I took during my undergraduate, like R-coding, bioinformatics for sure, evolution, and even my graduation thesis encouraged me to pursue a master's degree in this field. For instance, the topic of my graduation project was investigation of the microRNAs of selected autoimmune disease by bioinformatics. And Dr. Selin Küçükavşar, Selin Delicirmak, was my advisor in this graduation project. So of course, the methods related to bioinformatics that I used at the time, were quite simple compared to the ones I use now. However, at least it was a good experience for me, because, for example, I learned how to query different microRNA databases to gather relevant information and generating a phylogenetic tree for different organisms and analyzing them. So, analyzing and evaluating the results. So as a result, I have followed that path. The path I decided, being a bioinformatician. And now I'm preparing myself to take my last steps to reach my goal. A real bioinformatician. So if there are any questions that you want to ask, feel free to ask. I'd like to answer those questions. Otherwise, I would like to thank you for listening to me. So please stay healthy and strong, and Dankeschön. Thank you.
Thank you, Furkan, for sharing your story with us. And we don't have any questions for you right now. Okay. But for the participants, if you feel free, you can follow, as Furkan said, follow him on social media and ask him personally.
Definitely. So thank you very much again. Ciao.
Thank you for coming. Now we're gonna be with our last graduate presentation. It's gonna be Zeynep Hilal Durkaya. Welcome, Zeynep.
Hello, everyone. I hope you stay safe and good.
Welcome, Zeynep.
So I want to start to thank you all of my doctors and your organization committee for this symposium. I'm like the Furkan and Cansu. I'm 2019 grad students. So I want to introduce myself first of all. My name is Zeynep Hilal Durkaya, and I'm currently working on Akdeniz University Hospital Human Gene and Cell Therapy Center. I'm doing my master thesis, and also I'm a research assistant at here. So I want to start first of all how I chose this, what do you call it, like my journey. First years I was like not interested with that much the lectures or anything, and then I went second year. I was like, okay, I have to do something more. I need to more join everything. So I went to our doctors. I said, I want to be in the lab. I want to see what we are doing, what we will do. I want to see what's gonna happen. So I asked them if we have any projects on the lab I can attend. So on third year, Dr. Resime Hocam, she allowed to attend her TÜBİTAK project, and as a intern, we were intern. So I started at there, and then secondly, I went to microbiology department. I want to see other things, what we can do. I do my voluntary internship at there, and secondly, I went to pathology department. I did the internship at there also, and then I did my last internship in Akdeniz University Genetic Diagnosis Center. So, and then I realized the routine things like routine experiments is not like for me. I want to do more research. So then I went my currently advisor's laboratory and I talk with him. I say, I want to do research. I want to be more helpful. I don't want to be routine. So he offered me a position in his lab. So I started in his lab and my master degree. And then for the apply in Turkey, you have to do ALES exam and one YÖK allowed English exams. And you're attending a science exam in that university where you are applying. And then they're having an average of them. And then they're applying. If they say you are okay for our positions, they can accept you. So first year I was like, I couldn't manage all of them because we were attending also the lectures, medical faculty lectures with our advisor and dentistry faculty lectures. We will have to do help them and talk. So then after that, we had the TÜBİTAK projects for us. We start to do experiments, what we are doing. My advisor was studying on diabetes, and they had one project he developed on experiment animals about diabetes, type 2 diabetes on experimental animals. So I also attend experimental animal course because apparently I had to work with them. It was a kind of sad times because you have to, it's invasive techniques. So, and then I started working with experimental animals. My topic is neurobiology. I decided to on my third year. So I started working on what do you say? Amyotrophic lateral sclerosis diseases. So I did my projects on that. So I finished it. Now I'm my thesis year. But currently in my lab, they're working with the retinitis pigmentosa disease, COVID-19 vaccinating, and I think they continue the diabetes also. So what we are doing in our lab, I want the basic information give you. We are doing gene therapy. Basically, it's with viruses we are doing, lentiviruses or adenoviruses, FDA approved. So they had a article published on American Journal of Gene Therapy, the insulin treatment with GLP-1 showed. Yeah, now I'm continuing on my thesis experiments. It's about neurodegenerations. So I will continue with my PhD, but I offer all of you to more study on your bachelor degree because it's kind of important, your GPA generally, and what you achieved, what you learned, or understanding how mechanism working. For example, basically, we are talking about PCR nowadays, but what is PCR? We learned on bachelor degree. So it helps to understand the complex things because everything starts with step by step. Yeah, that's all. If you have any questions.
Thank you, Zeynep, for sharing your story. We don't have any questions. And I wish you good luck with your studies.
I hope everyone will do it.
Okay, so just a piece of advice for everyone. Just keep going. Don't stop. Follow your dreams, and you will reach it if you believe. Now, we're reached the end of our presentations, the end of our symposium. So I would like to offer our heartfelt thanks to all the lecturers who contributed their time to the symposium this weekend. I would like to thank all speakers, Dean of Students, the Department of Molecular Biology and Genetics, and all the participations, and the Genetic Student and EU Genetic Student Association staff. And please don't forget to follow us on Instagram, on the page, to check for our next events. We are now at the conclusion of our program and our symposium. Thank you so much for your participation, and we hope you enjoyed the presentations.
Catching a buzz.